This is one of my favorite lectures of the semester because it addresses a lot of misconceptions. Simply put, lactate is very misunderstood, and so the hope is that I can help clear up some of these misconceptions. So in order to do this, we're going to address the following 3 outcomes. Now, as discussed previously, whether or not pyruvate or lactate is formed at the end of glycolysis is dependent on the availability of oxygen. If oxygen is available, then pyruvate is formed. However, if oxygen isn't available, then lactate is formed. This can happen because the mitochondria can't move things into the mitochondria, and or there aren't enough mitochondria or mitochondrial enzymes to handle the products of glycolysis. What is also interesting is that different muscle fibers are more or less likely to produce lactate. Fast-twitched fibers have a type of enzyme or a type of lactate dehydrogenase, which is an enzyme that makes it easier for lactate to be formed as opposed to pyruvate. This shouldn't be surprising as fast-twitched fibers don't use much oxygen. So if they don't use much oxygen, they won't have a lot of oxygen available. Therefore, it's more likely that lactate will be formed. So don't let the title of this slide freak you out, lactate biochemistry. Let's just go back to glycolysis. If we have an excess amount of NADH, glycolytic activity slows down. Okay, this just means we don't have the ability to use glycolysis or for glycolysis to occur as quickly. Okay, in order to reduce the amount of NADH, we need to convert this NADH back to NAD. This occurs when hydrogen ion leaves NADH and binds to oxygen to form water. Okay, remember the electron transport chain? However, if oxygen isn't available, then the hydrogen ion from NADH will actually bind to pyruvate, which creates lactate. Okay, so lactate will take that hydrogen ion. So now we've got NAD, which is important and necessary for glycolysis to occur. Okay, so the human body does not appear to actually produce lactic acid, although admittedly there's some debate on this. So this leads to some confusion because the textbook says we produce lactic acid. In fact, many of you might be asking the question, don't we make lactic acid when we exercise? And the answer appears to be no. Okay, Dr. Robert Robergs of the University of New Mexico stated that the belief that lactate production releases a proton and causes acidosis or lactic acidosis is a construct and as such needs to be corrected. When it comes to lactate and lactic acid, it is one of the greatest controversies in modern exercise physiology and is one of the most entrenched constructs among both scientists and laypersons. Okay? And this is easy to see. Just go ask anybody at your gym. Go ask your trainer at the gym. What causes me to be sore? More often than not, they'll say lactic acid. Not true. Okay? And while I don't blame this necessarily on the people saying, I don't necessarily hold it against them that they don't know this. It's something that is completely incorrect. And so it's actually important to know lactate is beneficial. Okay, especially during exercise. So there are several reasons for this. First, lactate allows us to produce as much ATP as possible during glycolysis. A few slides ago, we talked about how we need to minimize the amount of NADH. High levels of NADH inhibit glycolysis. Well, by producing lactate, we're actually getting rid of NADH so that more NAD can be used to produce ATP. Similarly, lactate accepts hydrogen ions that are produced by glycolysis. That's actually what's happening when we're taking NADH and converting it to NAD. We're taking that hydrogen ion and forming lactate. This can also be true with just hydrogen ions floating around inside your cells. Ultimately, this reduces hydrogen ion concentration or acidity. In other words, lactate is a buffer. Finally, in order to get hydrogen ions out of the muscle requires the use of what's called a symport. A symport moves 2 or more compounds in the same direction, either into the cell or out of the cell. In this case, hydrogen and lactate both leave the cell through the symport. This reduces the acidity inside the cell. In essence, a single lactate molecule takes care of two hydrogen ions. One hydrogen molecule is taken up when we form lactate, and then a second hydrogen ion leaves the muscle cell when lactate leaves. So additionally, lactate can be used as a substrate or fuel source to produce ATP. In fact, there is some evidence that suggests that cardiac muscle cells prefer lactate as a fuel source. Similarly, lactate can be converted to glucose via a pathway called the Cori cycle. Okay, and so here we've got this diagram that shows the Cori cycle, and it works pretty simply. Lactate leaves the muscle cell, travels to the liver via the bloodstream. When it reaches the liver, it can be converted to pyruvate, and that pyruvate can ultimately be converted to glucose. Now, this process costs 6 ATP, but that glucose molecule can then travel in the bloodstream back to the muscle cell where it can be utilized in cellular respiration. Okay, so instead of producing 34, 36, 38 ATP, something like that, this will result in something like 28 total ATP being produced from a single molecule of glucose that goes through glycolysis, Krebs, and the electron transport chain. So I hope you have a better understanding of lactate. We'll obviously talk about this more in class, but before then, see if you can answer the following questions. Describe how lactate is formed, explain how lactate is beneficial, and see if you can summarize briefly how the Cori cycle works.